Hello, welcome back. We know that eukaryotic genomes are huge compared to that of prokaryotes. And we also have seen that the genomes of eukaryotes have a lot of DNA that we don't know what its function is yet, and if there is any function at all. So, and majority of it, if not all of it, comes from intronic regions. That is, eukaryotic genes are interrupted, and you call those sequences as introns. It is a huge burden to the cell to maintain these introns as genome, to transcribe them, and it's a metabolic and in a temporal ways to a burden. So there must be some kind of advantage to have these. Otherwise, why would evolution have these? That means there are metabolic disadvantages, but there must be something beneficial. Everything works by cost benefits in that particular circumstances the organism is living or existing. That is what we will try to understand. We'll try in these series of talks, we will try to see the chemistry of the removal of these introns, or we should say splicing. Many times we use splice out the introns. That is the wrong way to say it. Splicing literally would mean joining, and here we're talking about joining of exons. Exons are the coding regions and introns are the interrupting or intervening sequences. Welcome. Simply to have a good refresher on all the things that we need to understand, we will talk about introns as very simple, in a simplistic way though, that we have at the genomic DNA level, we have a promoter, and promoter is the region from which transcription would have initiated, and we would have a ribosome binding site. We will just call it this, which is essential for ribosome to recognize it and initiate translation, which is the process that would have followed splicing, RNA splicing. And here we have some exon 1, exon 2, and so on. Many of those are we have multiple exons. Here we are writing only 4 exons, but some of them can have more than 50 exons as well. And there are, which is because of having these introns, of course. Here we are representing 3 introns. So upon transcription, we would get a pre-mRNA or primary transcript, RNA transcript, and then we would call this region the 5' leader, and at the end we would also call this as the 3' or 3' untranslated region. Here we can call this as well as 5' untranslated region as well. So if we look into the exon, this is where the first exon would be having the start codon, and the last exon could be having the stop codon. That is where these things would be there. So and then each of these introns have a boundary like this, which we would recognize, or we would call them as 5' splice site, and at this side we would call them as 3' splice site. Here we are referring to the splice 5' to 3' of the intron. So each of the introns would be having this 5' to 3' splice site like this, and the machinery or the splicing machinery would splice the exons, which would mean that we are removing the introns as well. And the 5' splice site and 3' splice sites are recognized and eliminated, and in the process the exons are joined together. So now we have a mature mRNA. The mature mRNA has all the exons 1, 2, 3, 4 exons spliced together, and it has the 5' UTR and 3' UTR, which would also have the poly A signal here. Because of the poly A signal, we will also have poly A tail, and on this side we would be having, on the 5' side we would be having the G cap. That is a mature mRNA in eukaryotes. So that is what we will discuss, how the process of splicing of the exons happen. This is the step that we are trying to understand in this. We'll look at the chemistry, and then we'll look at the boundaries of the introns, and then we will go into the chemistry of it. The boundaries are actually very well conserved and based on each species at a time. Sometime, say, this is one of the examples that it's given, where here is an exon that we have, and this is the intron. All that is colored here is the intronic region, and then we have the exon. So we call them as 5' exon and 3' exon because our reference point is that of the intron. And again, at the same time, we'll also talk this junction as well as 5' splice site and 3' splice site, like we have discussed before. So there are certain conserved sequences at the 5' splice site as well as the 3' splice site. Say, for example, the exons mostly would end up with the penultimate base would be either C or A, and the ultimate base of the exon is usually G. And the start of the intron, that is the 5' splice site, would have sequences like G, U. In the third position, it could be either A or G, that is purines, and then A, G, and U. So that is the description of a 5' splice site. We don't have to memorize all of it, but it would be a good idea to at least remember that there are boundaries and there are conserved sequences within these boundaries of exon and introns. So that makes it easy for, or essential, that the splicing machinery could recognize them and then make precise cuts. Excuse me. Similar to 5' splice site, we also have 3' splice site. And in the 3' splice site, we have sequences something like this. We have N, that is, it could be any nucleotide, and cytosine, adenine, guanine. And the next exon would start with G. So all exons, at least within those that are in contact with the intron or adjacent to the introns, would start with G and end with G. That would make it easier for us to remember. And similarly, intron is also starting with G and ending with G. Let's look at, just to create a map so that we can try to remember by making statements such as these. So intron exon junctions would start and end with G. Say, for example, exon here is ending with G and intron is starting with G. And here intron is ending with G and exon is starting with G. At least that much we can remember. We don't have to really mug up all the base sequences here, but it's, as I said earlier, it'll be worthwhile to remember them, or at least recognize that there are these sequences. One another important thing is the branch site. Branch site is something within the introns, and they have Y and Y, which would mean Y stands for pyrimidine, PY. So Y is taken. Any of the pyrimidines is Y. There are several other conventions used. There are many other alphabets representing. Say, I have only taken those that are useful for our slide at this point. So Y is for pyrimidine, and it means either C or T. N recognize all bases. We don't have to really mention that. And R, pure. So R is taken for either purines, that is either A or G. So at least these three we can remember. So in the branch side, we have Y, which is for either of the pyrimidines, and N and Y. And then we have uracil. R is for any of the purines, and A and Y. One of the things that we need to remember when we are talking about this, say N, R, and Y, we also have for amino acids single letter codes. Y is for tyrosine, R is for arginine, and so on. So we shouldn't confuse between the amino acids and the bases that are being represented here. So the context is everything in these cases. So here is one important point, that is the adenine. And the adenine is the adenine that is present in this part is crucial for many of the things that happen during the splicing itself. This site itself is called as branch site, because that is where a new branch is going to form, which we will see later on. So the adenine has a crucial role to play when we talk for during the formation of or the splicing mechanisms. So in general mechanism is, as I was discussing, here we have an exon and an intron and an exon, and we need to get rid of the intron and we have to splice the exons together. So the branch site adenine that is present here has the important role. So the general feature is general mechanism is that the 2' OH, remember that we are talking about RNA. RNA has ribose as opposed to what DNA would be having. DNA would be having deoxyribose. The chemical difference being that in the second 2' carbon you have OH in RNA, whereas in case of deoxyribose we only have H. There is no O. So the 2' OH of this particular adenine is activated, and it would make a nucleophilic attack on the boundary of the exon intron on the 5' splice site, at the 5' splice site. So this is going to kind of bind here like this, or you can say it as this one binding here, and it forms a loop like this. This is called a 3-way junction. You have a nucleotide, and this is continuous, and you have a nucleotide, again a bond there. So the exon that is present here, 3' OH, at the end of the exon, is going to get activated and make a nucleophilic attack on the 5' side of the subsequent exon, and thereby exon 1 and exon 2 are spliced together, and whereas the intron is left out as a lariat. Lariat is structure that is formed like this is called as the lariat. I hope we have gained enough introduction to the splicing mechanisms. We will see chemistry in the later classes. Thank you very much.